Good morning, students. This is Prema, your biology teacher from BRGS. Welcome to my biology class. In this class, we are going to study about the heredity and evolution lesson. In the hereditary and evolution lesson, we should know about the definition of heredity. Okay, how it will occur, what is the meaning of heredity. Okay, the study of heredity and variation is known as genetics. We should know about the genetics first because the heredity comes through the genes. Yes or no? That's why we should know the meaning of genetics first. The study of heredity and variation is known as genetics. Heredity is defined as transmission of characteristics from parents to offsprings. Here offsprings means newborn, okay, or next generation. Newborn baby or the next generation, new individuals, okay. Hereditary is defined as transmission of characters from parents to offsprings. Here the characters are transferred from the parents to their offsprings, okay, or progeny. We call it as heredity. The differences in characters of parents and offsprings is known as variation. Here variation means the differences in character, it is not there in the parents, differ from the parents. New characters which is not belongs to the parents, that different characters we call it as variation here. Here there are two types of variations are there. How many? Two types of variations are there. Which are those? Somatic variation, gametic variation. Two types of variation. What do you mean by somatic variation? Somatic variation occurs in the somatic cells of the body. Here somatic cells means external body character. Okay, occurs in the external cell or in the body cells of the body. They are not inherited or transmitted in the next generation. These body characters not transmitted from the parents to their offsprings. Okay, this is not inherited from the parents. So they are also known as acquired character. Already it is there in our body. It is not inherited from the offsprings or parents or pre-existing cells. This is the acquired traits. Traits means character here. Next come to gametic variation. Gametic variation occurs in the germ cells of the body. Okay, germ cells means gametes. Okay, gamete cells of the body. They are inherited in the next generation. They are the inherited character means copied characters from the parents to their offsprings. Okay, so they are also known as inherited traits. These are the copying characters. That is not a copying character. Somatic cells are not the copying character, but gametic variations are the copying character, which is copied from the parents to their offsprings or the new progeny. This is about your gametic variation. Next, move on to the importance of variation. What are the importance for the variation? See here, the first point, it is the basis of the heredity. This is the basic unit of the heredity. Okay. Second one, it is the basis of the evolution also. See, we want evolution means they want the basic. The basic unit is variation here. To the variation only we can find the evolution of the different organisms here. Third point, that is it increases the chances of the survival of the organism according to the changing environment. Okay, even on environment change means the organism can adapt for the new environmental condition. These variations also help for the adaptation for the new environmental situations also. These are the importance of variations. Next come to causes of variation. What are the causes for the variations? Here the most common causes of variations are mutation. Here mutation means sudden changes in the chromosomes, number of chromosomes. That is called as mutation. Sudden changes. Okay. Recombination and random mating. Recombination means combination means repeat the copying character. Repetition in the copying characters also leads to the formation of variation, and also random mating. Mating means sexual contact. Random sexual contact also leads to the formation of variation here. Next point, that is recombination or crossing over is one of the important reason for variation. Recombination means repeating the copying characters, and also the crossing over. Characters are crossing over the other organism, or in between the two chromosomes. The characters will be exchanging. That we call it as crossing over here. That is also leads to the formation of variation or the varying characters. Okay, third point, that is it is a exchange of chromosome segment at the time of gamete formation. During the gamete formation, during X gametes or Y gamete, during the gamete formation, here the exchange of the chromosome segment. Okay, chromosome can be exchanged during the gamete formation. That is also leads to the formation of variation here. Next come to Mendel and his contribution in genetics. Okay, Gregor John Mendel started his work on Pisum sativum. Pisum sativum is the scientific name of the garden pea plant. Pisum sativum is the scientific name of the garden pea plant. Gregor John Mendel is the scientist who introduced the term genetics here. He was known as father of genetics. Who is the father of genetics here? Gregor John Mendel. He had chosen 7 pair of contrasting characters. Contrasting means opposite characters. Which are those? Here seed, gray and round color seeds, and white and wrinkled. Color of the seeds, yellow or green. In the flower color, white or violet. Pod, shape of the form, shape both are same. Full or constricted form. Color of the pod is yellow and green. Next, place of the stem, axial or terminal. Next, size of the stem, long or short. These are the 7 contrasting character observed in the garden pea plant. Scientific name of the garden pea plant is Pisum sativum. The reasons of choosing garden pea for experiment was, what are the reasons behind the choosing the garden pea plant? See here, the garden pea plant, they have short lifespan or life cycle. That means lifespan is less, means we will get more generation within the short time of period. Yes or no? That's why you chosen the garden pea plant for his experiment. Second, large number of seeds produced. Within a short period of time, they will produce more number of seeds. For experiment, we want more number of seeds within a short period of time. Yes or no? Next, third, that is self-pollination. We can make the self-pollination in the laboratory condition also. We can take new generation easily. Okay. Next, several contrasting characters can be found, or several contrasting characters means we can find the more number of characters within a single plant. We will take other plant, we will take only 1 or 2 characters, but in the garden pea plant we can find more number of contrasting characters in the same plant. That's why he chosen garden pea plant for his experiment. This is very important for your examination also. Next, come to Mendel laws. See, Mendel introduced so many laws about the genetics or in the on his experiments. First one, that is law of dominance. If the 2 alleles at a locus, if the 2 alleles means 2 chromosomes are present on the same chromosome or in the same place, then the dominant allele determines the organism appearance. One gene refers to the characteristics of the main characteristics of the plant. For example, capital T represent tallness character of the plant. Okay, small t represent the dwarfness character of the plant. Okay, your capital after gamete formation, In the F1 generation, we will get capital T and small t combination. Here, the capital T always represent the dominant character, while small t is present also, they will express their character, that is tallness in the plant. Yes or no? Like that, the first or one allele or one gene represent the dominant character, means original character of the plant, that is called as dominant character. Okay, another present small t that represents the recessive character of the plant body. When the dominant capital T is present along with the small t, they represent only the dominant character, because here the dominant character which suppress the recessive character of the plant. We are not telling this is recessive plant or in the dwarf plant. We are always telling the dominant character of the plant body. Okay, this is about your law of dominance. Always we are telling the dominant character of the organism or the plant. When the recessive character also present along with the dominant character also, we are telling only the dominant character. Next come to law of segregation. Segregation means separation, during separation. Okay, the two alleles for an inherited character are separate during gamete formation and end up in different gametes during the gamete formation, that is during capital T and small t, that gives capital T one gamete from the capital T combination. In the small t combination, they will give only small t gamete. During the gamete formation, they are separate. Capital T separate, small t separate. Again, the end up in the F1 generation, again combination, we will get capital T, small t. Okay, this is end up with the different gametes. Next, third law, that is law of independent assortment. Each pair of alleles segregates independently of each of other pairs of alleles during gamete formation. See here, each pair of alleles means two pair of alleles, capital T, capital, like that. We have two alleles segregates independently. We are separating the gamete separately of the other pairs of alleles during gamete form. During the gamete formation, we are segregating or assorting the similar kind of gametes. That is the law of independent assortment. This is the laws of Mendel's, very important for our examination. First law, that is first hybridization, that is monohybrid cross. Monohybrid, here name itself indicates the definition. Mono means single, hybridization means mixing of the characters. Okay, when one pair of contrasting character was taken to cross two pea plants, we are taking two single character here. The crossing between the two parents which are differing in the single contrasting character. Crossing between the two parents which are differing in the single character, differing in only single character. Okay, that we call it as monohybrid cross. See here, we are taking true breeding yellow pod and true breeding green pod. Only single contrasting character here, green and yellow pod. See, after crossing we will get capital G, small g. Here capital G represents the dominant character, small g represents the recessive character. Here the dominant character is the yellow pod, recessive character is the green pod. Okay, this is the monohybrid cross. That picture or chart represents the monohybrid cross between the true breeding yellow pod and true breeding green pod. All the pods obtained was green in color. We know that already, capital G, small g. Okay, the offsprings obtained are known as F1 progeny or first filial progeny. In the first hybridization, we can find as F1 generation, that is first filial generation. In case of heterozygous condition, we are getting we studied in the only homozygous condition, but here in the heterozygous condition, the parents are heterozygous means two different means capital T means homologous condition, homozygous condition. Capital T small t is the heterozygous condition. Parents are heterozygous in condition means capital B and small b is the heterozygous condition. So phenotypically three purple flower and one white flower was produced, but genotypically one homozygous dominant, that is capital B. See in the chart, capital B, small b. We are taking for the crossing. See, first column represent the capital B. Here both genes are similar, capital B only. That will be called as homozygous flower. Okay, homozygous flower. That is white flower. Okay, capital B, small b, it is the heterozygous in condition that leads to the formation of purple flower. Okay, we will get only one white flower. All the three flowers are purple in color because here only small b is the homozygous white in nature. This is about monohybrid cross in the heterozygous in condition. Next come to the dihybrid cross. When two pairs of contrasting characters are taken to cross, we are taking two contrasting character here. These are difference in the two contrasting character between the two parents. Okay, between the two parents, it is known as dihybrid cross. See here we are taking yellow round and green wrinkled. Yellow, green is the one character, round and wrinkled is the second character. Okay. After crossing between the yellow round and green wrinkled, we will get F1 generation, we will get yellow round only, but mixturing of the character. See here, capital Y, capital R, small r only. But here we are telling only the dominant character, yes or no? That's why we call it as yellow round in the first generation. After crossing after self crossing, again we will take, see the gametes after the F1 generation, we will get capital R, capital Y, capital Y, small y, capital R, small y, small r. Same, we are taking two sides are similar gametes. After we crossed, we will get this ratio. The phenotypic ratio was found to be 9:3:3:1. Here the 9 are round yellow in color, 3 are round green in color, 3 are wrinkled yellow in color, one is the wrinkled green in color. But genotypically we are finding out the 1:2, 1:2, 4:2, 1:2, 1. This is the genotypic character. We are telling while experiment genetically. Okay, that we call genotypic ratio. Phenotypic ratio means external appearance of the plants. We are counting external character. But in the genotypic ratio, we are counting the genetic information. That means in the genes. Okay. Thank you for your kind attention. We will continue in the next class.